Okay, guys, it's 3 minutes past, and if anyone wants to join coming in late, they can. We can get going. Just to introduce myself, whoever his first time it is here, my name is David. I am the distributor manager here at Assay Genie, and my role within the company is to train our distributors on any of our current or existing products, or any new products that we have coming down the pipeline too. And it's my great pleasure to be here with all of you today to speak to you about our biosimilar product range. And today I'll be talking through how we can advance translational research with in vivo biosimilars and reference antibodies. And in this presentation, I'll speak to you about our VivoGenie in vivo biosimilars, our reference and conjugated antibodies in standard, functional grade, low endotoxin, and FC mutated formats. And this is the agenda for today. I'll talk to you a little bit about us and who we are as a company. I'll give you an overview of biosimilars and the history around immunotherapy. I'll speak to you extensively about our VivoGenie biosimilars and our reference and conjugated biosimilars. And then I will finish with a word of thanks. And for those of you who are unfamiliar with Assay Genie and whose first time it is here today, Assay Genie was actually co-founded in 2017 by our co-founders, Dr. Cullum Ryan and Dr. Sean McFarrig on the right-hand side. We're based and headquartered in London, but our primary base of operations, where we QC all of our products, is in Dublin, in the Republic of Ireland. We have distributors in over 80 countries and over 1,000 product citations. And all of our products are ISO 9001 certified. And we work with and collaborate with some of the world's leading biopharmaceutical and academic institutions too. And this is a great snapshot overview of the 5 key and core technologies that we offer at Assay Genie. We offer over 32,000 different ELISA kits, over 11,000 highly pure and recombinant proteins. We have an amazing collection of different multiplex assays that rivals our competitor Luminex. We have an amazing collection of different biochemical and activity and cell-based assays, and we can't forget about our antibody range, which I'll be talking to you about extensively today. And this is our company's mission, which we strive to and adhere to every single day we work, which is to make scientists smile through using our innovative products, our fun and quirky marketing with our smiling scientists and Genie logos. And what we pride ourselves on at Assay Genie here is our remarkable interactions with our team. And these interactions include technical support that is done by either email, video, or phone, or by our live chat, which is manned by one of our colleagues from our team. And then we offer extensive troubleshooting and technical support. And if anyone is actually ever unhappy with any of our kits, we do also offer a replacement kit. And we also do offer a money back guarantee too as well. But we hope that with all of the support that we offer, that it doesn't get to that step too. And now, I'd like to actually just speak to you and talk to you through the historical perspective of therapeutic antibodies and really how we got to where we are today before I actually speak to you at length about the science behind them. And what I want to first do is actually define what a biosimilar is. And by the FDA definition, a biosimilar antibody is an FDA approved biologic that is highly similar to its reference biologic with no clinically meaningful differences despite not being an exact copy. Biosimilars can be chimeric monoclonal antibodies with murine variable regions and human Fc immunoglobulins to reduce immunogenicity, but biosimilars can also be humanized monoclonal antibodies that are murine originally but designed to be identical to a human antibody. They can be identified by certain suffixes such as OMAB, ZMAB, Zumab, and UMAB, and as of 2024, there has been 66 FDA-approved biosimilars. And when we discuss the history of immunotherapy, we first have to actually go back to the 1890s when the first cancer vaccine was developed by William Coley. Fast forward a few years back into 1975, we had then the birth of hybridoma technology, and in 1978, we had the discovery of tumor-specific monoclonal antibodies. And then there was the key discovery in the 1990s of the checkpoint inhibitor by Professor James Allison and by Professor Tasuku Honjo, and I'll mention them briefly in a short while. And then in 2002, we had actually the first biosimilar being approved, which was called rituximab by the FDA. And then in 2009, we had filgrastim, which was the first FDA-approved biosimilar for cancer treatment. And when we talk about actually immune system modulation and the history of immunotherapy, there's been no two greater figures in the fields of immunology that have made a greater contribution than these two individuals right here. This is Professor James Allison from the University of Texas on the left-hand side here, and then this is Professor Tsuko Honjo from the University of Kyoto. And both of these individuals were co-awarded the Nobel Prize in Physiology and Medicine back in 2018 for their key and seminal work on anti-PD-1 and anti-CTLA-4 antibodies, which have helped to accelerate discoveries in the fields of cancer, in autoimmunity, and neurodegeneration, and other diseases too. And when we actually talk about their work, this image is a great representation of the work that they did. And here you can see in green, these are T cells. And here we can see in pink, these are then tumor cells. And our T cells express these specific receptors called PD-1, whereas our tumor cells express these other receptors called PDL1. And when these two receptors actually interact and bind together, this causes a deactivation of T cell activity. However, what Professor Allison and Professor Honjo discovered was that if you block both of these receptors with either anti-PDL1 or anti-PD1 antibodies, this causes an increase in activation of T cell activity, which then ultimately leads to increased tumor cell death. And when we talk actually about the original drugs versus newer versions of biosimilars, there's extensive key features which have changed since the original drugs have come out. When we look at the structure and engineering of the original biosimilars that were developed, these were humanized or fully human monoclonal antibodies used to block PD-1 interactions with either PDL1 or PDL2. Now, with the newer versions, these next generation antibodies are also bispecific antibodies and have FC engineered variants for enhanced function. When we look at the original biosimilars binding and efficacy, they had standard binding affinity to PD-1 requiring consistent dosing, but now the new modern and newer versions have stronger PD-1 binding, longer half-lives, and improved immune activation. And when we think of the delivery and formulations for the original biosimilars, they were mainly administered through intravenous administration, but now these newer versions include subcutaneous formulations for easier administration too as well. And we cannot actually forget about actually the features of bispecific and multi-specific antibodies, where original biosimilars, they were monospecific that only targeted single and individual targets. Now with these newer biosimilars on the markets, these are actually bispecific or multi-specific antibodies, and examples of this include, say, targeting PD-1 and CTLA-4 together, or targeting then PD-1 or LAG-3 together for enhanced immune responses. And finally, when we look at the costs of these original drugs, these were very expensive due to patents which limited biosimilar availability. But with the newer versions on the markets, biosimilars are being developed for cost-effective alternatives. And now I want to just talk to you about some of the market challenges and future perspectives for biosimilar antibodies. And when we actually look at the market and having done a lot of analysis on the market, we can see that the market size is expected to reach 1.5 trillion by 2033. And there's going to be a huge market to take from that too. And Assay Genie wants to be a world leader in providing and producing biosimilars for academic and biopharmaceutical labs around the world. And where we see biosimilars being positively used includes the fields of oncology, growth hormonal deficiency research, in blood disorder research, and in chronic and autoimmune diseases and infectious diseases research too. And There's three major problems in drug discovery in this current time. The first issue is in vivo preclinical issues, and many research biosimilar antibodies have high endotoxin levels, which lead to off-target immune activation in animal models, and most antibodies are not in vivo grade, causing failed preclinical studies and a waste of resources and time. The second problem is that there's a lot of biosimilar development and regulatory hurdles, and pharma companies now must demonstrate equivalence to current market antibodies requiring high quality reference biosimilars. And without reference and validated biosimilars, regulatory approval is delayed, costly, or even denied. Thirdly, and lastly, is that targeted therapies need better drug delivery solutions, and conjugated biosimilars are absolutely critical for antibody drug conjugates, for imaging, and for targeted therapies. And lack of reliable conjugated biosimilars slows down drug discovery and therapeutic advancements. And our solution here at Assay Genie is that we are offering over 1,000 biosimilars to help accelerate this discovery and to get rid of these problems. In our VivoGenie biosimilar range, we have over 100 VivoGenie biosimilars, which are formulated for in vivo applications, ensuring safety and efficacy in preclinical animal studies. We have over 600 reference biosimilars designed to be nearly identical to an original biologic medicine used in research or in preclinical studies too. We have over 658 biosimilars labeled with fluorescent dyes or enzymes for enhanced detection and quantification, and we also have quantitative and qualitative free drug and ADA ELISA kits for biologics and biosimilars, which we will be doing another webinar on. And now I actually want to speak to you a little bit more about our specific VivoGenie biosimilar antibodies. And these are functional grade biosimilars for in vivo preclinical animal studies. And I want to ask you the question, which is when are in vivo biosimilars needed? Well, very simply, they're needed to and used in to model disease states in translational research, in specific cell line derived xenograft mouse models, in orthotopic mouse models, or even in patient derived xenograft mouse models. And every single one of these models can be treated with immune checkpoint monoclonal antibodies or even antibody mediated cell depletion to modulate the immune response. And our biosimilars and biosimilars in general, right, are essential for disease research. On the left-hand side here, we have a number of different biosimilar therapies, including T-cell depletion, which is used in disease areas such as transplantation, autoimmunity, leukemia, and obesity. And then we also have biosimilars for autoimmune diseases, including rheumatoid arthritis, psoriasis, irritable bowel disorder, and ankylosing spondylitis. Finally, then we also have biosimilars for cancer, which is used in the likes of non-Hodgkin's lymphoma, breast, and also colorectal and gastric cancer too. And there are three major challenges currently facing in vivo scientists. The first one is endotoxin contamination. High endotoxin levels trigger immune responses, which affect and negatively impact study reliability. Number two, there's inconsistent batch-to-batch variability, which leads to unreliable pharmacokinetics and efficacy. Thirdly, and lastly, is that around the world and globally, there's actual lack of in vivo validated biosimilars, and many of these biosimilars are actually not optimized for animal models, reducing study success. And at Assay Genie, we offer over 100 of these different VivoGenie biosimilars. Some of our top biosimilars include the likes of pembrolizumab, atezolizumab, anifrolumab, and also rituximab. And these can be used in multiple different research fields, including the likes of cancer immunotherapy, and autoimmunity, and also in inflammatory diseases and infectious diseases too. And all of our VivoGenie biosimilars are CGMP grade and are manufactured in ISO 9001 accredited facilities. Each and every one of our biosimilars has little to no immunogenicity, and also all of our biosimilars are low endotoxin too, which are perfect for in vivo mouse or animal studies. And our biosimilars also come in FC reduced options. And they are tested in a wide range of different applications, including in preclinical animal studies, like I've mentioned, in cell depletion studies, in blocking studies, in neutralization studies, and in co-stimulation studies too, and a wide number of other research and experimental design studies. And these are some of our top biosimilars which have been used in research use only settings, including, like I mentioned, pembrolizumab, which targets anti-PD-1, atezolizumab, which targets anti-PD-L1, and anifrolumab, which covers and targets anti-interferon-1, and also rituximab, which targets anti-CD20. And also, at Assay Genie, we pride ourselves on offering over 25 unique biosimilars, which no other company offers, including alemtuzumab, ibalizumab, and also natuzumab as well. And these target anti-CD25, anti-CD4, and also anti-EGFR2. And in each of our biosimilars, we have industry-leading lot-to-lot consistency. And here I'm just using our in vivo PD-1 antibodies as an example. And here you can see we have endotoxin analysis and for each of our different lots of our PD-1 antibodies. And here you can see they are less than or equal to 1 endotoxin units per milligram for all of our 8 different lots. And we also undertake chromatographic analysis too, and we also undertake reducing and also non-reducing SDS page to demonstrate and to show all of you that we have consistently high purity lots amongst every single lot of our antibodies that we produce. And now I just want to talk you through some of our case studies where our antibodies and our biosimilars have actually been used in the field. And I want to share with you a success story about our in vivo antibodies, our anti-PD-1 and anti-CTLA-4 antibodies, which have been manufactured in the same way and to the same high manufacturing standards as our biosimilars have been. And what we did was we got an independent CRO to analyze the impact of our different antibodies, our anti-PD-1, PD-L1, and anti-CTLA-4 against one of our competitors that also has the same matching antibodies, clones, and also isotype controls. And when they did this, they did it in a CT26 syngeneic colorectal carcinoma mouse model. And over 26 days, they evaluated the impact of our in vivo antibodies against our competitor. And here, when you look at each of the graphs, you can see that in the red and in the purple, the isotype control did not actually reduce the size of the tumor as expected. But here you can see that our different clones, our RPM1-14 and our clone 29F1A12, significantly and more efficaciously reduced the size of the tumor than our competitor. And this was to the same effect as our CTLA4 in vivo antibody, which helped to significantly reduce tumor size over 26 days. It is worth noting that our competitor's antibody also reduced tumor size, but not to the same effect and precision as our Assay Genie antibody. And this is important because our Assay Genie antibodies offer superior reduction qualities with our in vivo functional grade anti-PD-1 and CTLA-4 antibodies. These antibodies achieved significantly greater tumor size reduction after 26 days compared to our competitor, Vardner B. What we want to do next is we want to enhance checkpoint blockade strategies to further improve tumor regression and survival outcomes in colorectal and other cancers. And scientists are continuing to rely on our in vivo antibodies for cutting-edge immuno-oncology research. And a case study I wanted to share with you was when a specific biotech company used our pembrolizumab and our atezolizumab biosimilar antibodies. And the challenge here was that they wanted to perform in vivo experiments targeting macrophages in two different labs, one based in China and the other based in the United States, and they needed high quality in vivo grade biosimilars. Our solution, when we were speaking to them, is that we wanted to offer our VivoGenie pembrolizumab and atezolizumab for its high purity, which is greater than 95% in purity, low endotoxin levels, which is less than 1 endotoxin unit per milligram, and also low immunogenicity. We offered our high quality pre-sale sample specific support, fast delivery, and optimal pricing, and they found that the results were highly reproducible due to the high quality of our VivoGenie biosimilars. And another success story I wanted to share with you just to show the impact of our Assay Genie products is the impact of some of our in vivo antibodies. And here, with the collaboration between Professor Ed C. Lavelle and Professor Mats Andersons from Trinity College Dublin and the University of Florida, respectively, they actually showed through using our antibodies that our antibodies can enhance survival and reduce tumor growth. And this was recently published in Cell Reports Medicine in May of last year. And when they approached us with a specific challenge, they found that STING targeted therapies, STING standing for stimulator of interferon genes, that these therapies they failed clinically due to poor cellular uptake, excessive inflammation, and weak tumor immune responses. When we spoke to them at length about what they needed, we found that our in vivo CD8 and natural killer 1.1 monoclonal antibodies would be perfect to determine what immune cells were required for the anti-tumor effects of C100, which is a STING activating adjuvant. We supported them in optimizing the dose response for both of these antibodies in the depletion studies. And what they found is that by recruiting CD8 positive T cells into tumors, C100 sensitizes tumors to PD1 blockade, improving immunotherapy success. This was the experimental design and the experimental setup right here, and mice were injected intraperitoneally with an anti-CD8 alpha antibody and an anti-natural killer 1.1 antibodies, both of which were provided by Assay Genie. And then this was followed up by further injections on the respective antibodies every 4 days throughout the study. And you can see here that the frequency of CD8 positive T cells among total CD8 3 positive T cells were measured 7 days post-injection, and here you can see that they are no longer expressing CD8 due to our antibody. Similarly, right here, you can see that there's total natural killer cell depletion 3 days post-injection, demonstrating that both of our antibodies were working effectively. And when you looked at the results a little bit more, you can see that there was total CD8 depletion, tumor size reduction, and overall survival in the animal model. They did this by actually, number one, by looking at the tumor growth. And then when they looked at the C100 treated mice, they can see that tumor growth was significantly inhibited here in orange. But depletion of CD8 positive T cells, which is the orange and white right here, abolished this effect. This is super important as it demonstrates that CD8 positive T cells are critical and very important mediators of C100 induced tumor suppression. They also did a Kaplan-Meier survival analysis, and they found that C100 treatment alone, or in combination with anti-PD1 here in orange and here in purple, significantly prolonged survival. But CD8 positive T cell depletion here in orange and white completely reversed this survival benefit. And this is very important because C100 provides a breakthrough STING targeting strategy that enhances tumor immunity while minimizing inflammation. And preclinical scientists can optimize tumor reduction and survival by precisely depleting CD8 positive T cells and natural killer cells using our low endotoxin functional grade in vivo antibodies. And what's next? And what's coming down the pipeline is that C100 is now being explored for clinical translation in combination with different immunotherapies. And scientists are now continuing to rely on our functional grade in vivo antibodies to help advance cancer immunology. And just to summarize our VivoGenie biosimilars, these are in vivo grade biosimilars, which are key to development of new immunotherapies. They are of high purity, low endotoxin, and are needed both for in vivo and in vitro research. And very importantly, our VivoGenie biosimilars have endotoxin levels less than 1 endotoxin unit per milligram and greater than 95% purity. And the VivoGenie range guarantees reproducible results in all applications, including in different functional assays, in ELISA experiments, in PK and PD assays, also in Western blot and immunofluorescence experiments and flow cytometry experiments too. Okay, guys, as we're coming to the end of this webinar, I'm going to be just talking to you now about our reference and conjugated biosimilar antibodies. And these are purely research-grade biosimilars for in vitro studies. And when you differentiate between both reference and conjugated biosimilars, you first need to look at their purpose. Reference biosimilars are used for functional comparability to reference biologics, whereas conjugated biosimilars are mainly used for detection and quantification of specific targets. Reference biosimilars are non-conjugated, whereas conjugated biosimilars are attached and labeled with fluorescent labels such as biotin, such as APC, phycoerythrin, or FITC. And when we look at the applications of both of these different biosimilars, both of them can be used in vitro experiments, whereas reference biosimilars can be used in the likes of Western blot experiments or in ELISA experiments. Or conjugated biosimilars can be used in the likes of PK and ADA assays and flow cytometry experiments, but can also be used in ELISA experiments too. But very importantly, both our reference and conjugated biosimilars can be used in pharma and pharmacokinetic research, in drug development, and in cell therapy research too. And we have over 1,200 reference and conjugated biosimilars. Like I mentioned, some of our top biosimilars include pembrolizumab, atezolizumab, anifrolumab, tocilizumab, and also rituximab too. And the research fields where this can be used include pharma and pharmacokinetics, drug development, and also in cell therapy research too. Just like with our VivoGenie biosimilars, our reference and conjugated biosimilars are of premium quality and are manufactured in ISO-accredited 9001 facilities. Like our VivoGenie biosimilars, these have little to no immunogenicity and can be used in vitro experiments, which are validated for a number and wide different of applications, and also are designed to be nearly identical to the original biologic medicine and highly specific to the target of interest. And this is just a list of some of the unique and validated biosimilar antibodies we have in our portfolio. I'll let you guys read through this list in your own time to see what unique targets work for you. And the tested applications where our reference and conjugated biosimilars have been used, like I said, include ELISA, PK, and ADA assays, in flow cytometry experiments, in Western blot experiments, in immunohistochemical experiments, and also in surface plasmon and resonance experiments, which I will mention briefly. And this is some of the extensive validated data that comes with our reference and conjugated biosimilars. Here on the left-hand side, you can see some reference and validated data for ELISA experiments. And on the right-hand side here, you can see some of the validated data for flow cytometry experiments too. And another major application where reference and conjugated biosimilars can be used is in surface plasmon resonance, which is an optical label-free detection technique used for the real-time in situ investigation of reversible biomolecular interactions, such as the specificity of an interaction, the binding affinity and binding levels, and also can be used to quantify the dissociation and association rate constants too. And there is a major need for biosimilars to quantify and to measure anti-drug antibodies. And these anti-drug antibodies measure the influence of the pharmacokinetics of the drug and are associated with reduced clinical efficacy and increased risk of adverse events such as infusion reactions. And one case study I wanted to actually share with you was when the Fraunhofer Institutes used our reference biosimilars. And when they were using it, they wanted to perform an in vitro experiment for analytical comparability and needed a reference biosimilar as a biosit of control. So they used their anti-VEGF biosimilar. And when they came to us, we thought that our anti-VEGF biosimilar would be perfect as it was validated for SBR and in different bioassay applications. We offered our high quality pre-sale support. And when they came back to us with the results, they found that our biosimilar worked excellently and in a biocore SPR measurement and in a VEGF bioassay. And in terms of activity, it was comparable to the reference values provided by the manufacturer, which was excellent to see. And just a final reference biosimilar example I wanted to share with you was when a pharma company came to us and they wanted to evaluate the different bioactivities in an in vitro assay with cell cultures. And this lab was investigating the functional activity of a reference biosimilar for a therapeutic monoclonal antibody in a cell-based assay. And the goal here was to assess its ability to blind the receptor and activate signaling pathways. We provided one of our reference biosimilars for them to validate, which was already highly validated and we knew would work for what they needed it for. And when they came back to us, they showed that the study confirmed that the reference biosimilar effectively mimicked the original biologic in receptor binding and in downstream pathway activation. And this meant that the biosimilar could be used as a reliable and effective control. So, just to really summarize what I just spoke about right here over the last half an hour, reference biosimilars and conjugates play a crucial role in translational research by bridging the gap between preclinical findings and in clinical applications. Reference biosimilars are standardized antibodies that serve to compare new biological products, ensuring assay reproducibility and being more affordable. And conjugated biosimilars allow for precise monitoring of drug target interactions, immune responses, and biomarker expression, facilitating translational research. And finally, Assay Genie's biosimilars are validated for head to head studies and in vitro applications and facilitate the investigation of new therapeutic antibodies. I'd like to thank everyone for coming on this call today in such short notice. And if anyone does have any questions in the group chat, please fire them away and I'll answer them now. But please also reach out to us either at distributors@assaygenie.com or feel free to set up a call with us or even just email info@assaygenie.com or ask some questions on our live chat support and I'll happily take them. This presentation has been recorded too, and then we can send out and share the recording with you afterwards. So thank you all so much for coming to the call. Does anyone have any questions? No problem at all, Joe. Thanks for hopping on the call. No worries at all, Ilka. My absolute pleasure. No worries, Wasim. Rania, you have a question? Fire ahead, please. Yeah, that's a great question there, Anya. Endotoxin contamination can just usually come just from actually the in vitro experiments anyways. And so usually it can come from just a number of different paradigms and experiments. And so really we try to control for that as much as possible. When you look at Assay Genie's endotoxin levels, they're some of the lowest around, which is what's very popular with some of our customers. And so it just demonstrates, and we continually demonstrate consistent high purity amongst all of our lots. I hope that answered the question. No worries at all. Does anyone else have any other questions, guys, before we head off? Yeah, that's a great question from you, Kirthika. Yeah, no, there has been actually no studies have actually shown any rejection for our biosimilars. We do extensive testing on all of our biosimilars to really show that they do work and that they work extensively too. And so really when we have these biosimilars and we release them, we always make sure that they are working effectively in all studies. Yeah, Kandika, I'll absolutely share the PDF to the Biotechno Labs team. So I'll send that on to Elizabeth or Priya, and then you should receive that by the end of the day. Hey Danielle, that's a great question right there. I actually wouldn't have the exact answer to that right now, but if I can just go actually speak to our lab about that, I'd be able to actually come back to you with a specific answer for that, if that's all right. No worries at all. I'm just going to take note of this right here, and then I'll go up and then I'll have a discussion with our lab just to see how that can be done. Yeah. Okay. And guys, before we finish off, does anyone have any last questions? I don't think so. I don't think there is. And as I said, if there ever is any questions, you know where to reach me, you know where to give me a call. And thank you all so much for joining me. And yeah, I'm looking forward to seeing everyone tomorrow for our Q2 promotion launch as well. So I hope everyone has a great rest of your day, and we'll be in touch soon. And sorry, Candiga, I'm just after seeing your comment right there. Lead time, delivery time, it's always a standard, 7 to 10 days. But some of our biosimilars, they do have to be actually custom made. So usually that does take a little while longer as well. So the production might take maybe 5 days for some of our biosimilars, and then it'll be maybe 7 to 10 days for delivery on top of that. But usually we do have some of them in stock, so the lead time is 7 to 10 days. Okay, guys. I'm going to log off right now. Thank you again so much for coming to this. It was great to see all of you and have a great rest of your day. Take care. Bye bye.